Welcome to Learn While Sleeping. This is the podcast where you can drift into rest, where your mind explores the quiet wonders of science. Each episode is a calm lecture, slow and steady, meant to teach and soothe at the same time. You don't need to stay awake to follow along. Your body can rest, and your thoughts can soften, and the science will simply flow around you. If this is your first time listening, you are invited to let go of the day. Find a comfortable position. Let your shoulders settle. Take a slow, deep breath in, and gently release it. Here, learning is not rushed. It's not stressful. It is gentle, like reading by the candlelight, or listening to a quiet story by the fire. Take another slow, steady breath, and let yourself drift into calmness. Tonight, we begin a journey into the story of the immune system, not as a dry lecture, but as an interesting exploration. Imagine walking into a grand hall of history, where the first discoveries of immunity are written on the walls. Our journey begins with Edward Jenner, an English physician in the late 1700s. Picture him carefully observing farmers and milkmaids. He noticed that those who had once been infected with cowpox, a mild illness spread from cows, seemed immune to the far more deadly disease, smallpox. In 1796, Jenner performed a gentle but bold experiment. He took material from a cowpox blister and introduced it into the arm of a young boy, James Phipps. The boy became mildly ill, but when Jenner later exposed him to smallpox, he did not fall sick. It was the first successful vaccination. Vaccination, a word that comes from vacca, the Latin for cow. Jenner is remembered as the father of immunology, his careful act of protection marking the beginning of a new science. Years later, another scientist, Louis Pasteur, extended Jenner's ideas. Pasteur experimented with microbes themselves, discovering that if you cultured them under weakened conditions, they lost their ability to cause severe disease, but could still train the immune system. This phenomenon was called attenuation. With this approach, Pasteur developed vaccines for rabies and anthrax in the 1800s, saving countless lives. Around the same time, Robert Koch laid the foundation of bacteriology. He was the first to show that specific microbes cause specific diseases. Tuberculosis, anthrax, cholera. Koch isolated each one, linking microbes to illness with a precision that confirmed germ theory. He gave immunology a scientific grounding, proving that microbes were real enemies and the body had to fight. Decades later, the world witnessed one of immunology's greatest triumphs: the eradication of smallpox. Can you believe it? Through widespread vaccination, the World Health Organization declared in 1979 that smallpox had been completely wiped from the earth. It remains the only human disease we have ever fully eradicated. Proof that the immune system, once trained, can overcome even the deadliest foes. As you rest, imagine these pioneers: Jenner with his observation, Pasteur with his careful experiments, Koch with his microscope and cultures. Their work opened the first pages of immunology, and their discoveries echo in every vaccine and every protected life today. Let's take a break and try to reflect on this. Part 2. Immunology in Medicine. Now, let us leave the halls of history and step into the present. The science of immunology is no longer about smallpox or rabies. It touches nearly every corner of modern medicine. Imagine 5 great doorways, each leading to a different landscape where the immune system plays a central role. The first doorway opens into the world of allergy. Here, the immune system, instead of protecting, becomes overly sensitive. Harmless substances like pollen or dust are treated as invaders. The result is sneezing, itchy eyes, inflamed skin. In some cases, like contact with poison ivy, the skin erupts into a rash as immune cells flood the area. Allergies remind us that the immune system's strength must always be balanced with restraint. The second doorway leads to autoimmunity. Here, the immune system becomes confused, mistaking the body's own cells for foreign enemies. For example, in rheumatoid arthritis, joints swell painfully. In type 1 diabetes, the immune system destroys the insulin-producing cells of the pancreas. In multiple sclerosis, it attacks the protective sheaths of the nerves, otherwise known as myelin. These conditions reveal how devastating it can be when the immune system turns against the self. The third doorway reveals transplantation. Imagine surgeons carefully placing a donor kidney into a recipient. Well, the body's immune system, trained to distinguish self from non-self, sees the new organ as foreign. Without careful management, the immune system will attack the graft, seeing it as a threat. The story of transplantation medicine, beginning with the first successful kidney transplant in 1954, has been the story of learning how to calm the immune system enough to accept life-saving gifts. The fourth doorway opens into cancer immunology. The immune system has the power to recognize cells that grow out of control. Yet, cancers are skilled at hiding. In recent decades, scientists have developed immunotherapies, treatments that help the immune system see and attack cancer. One good example is monoclonal antibodies, immune checkpoint inhibitors, and CAR T cells. These are among the fastest growing and most promising therapies in medicine today. Finally, the fifth doorway leads into infectious disease. From influenza to HIV, from malaria to COVID-19, microbes continue to test the immune system. Some evolve quickly, staying one step ahead of our defenses. Vaccines and therapies remain our greatest tools, but the immune system is always the first line, meeting pathogens at the moment of invasion. Together, these 5 doorways, allergy, autoimmunity, transplantation, cancer, and infectious disease, show the vast reach of immunology. As you breathe slowly, imagine walking past each doorway, just glimpsing the ways immunity shapes health and disease, taking a peek in each one. The immune system is not only a defender against microbes, but a central player in nearly every field of medicine. Let's take another break to reflect on this. Part 3. Pathogens and Microbiota. Imagine now that you are standing at a crossroads. On one side are the countless microorganisms that threaten health, the pathogens. On the other side are the equally countless microbes that live with us peacefully, even helping us thrive. The immune system must constantly distinguish between friend and foe. Pathogens can come in many forms. There are bacteria, some causing illness like tuberculosis or cholera, while others live harmlessly in the gut. There are viruses, tiny packages of genetic material that invade living cells to replicate. There are fungi, from common yeast to dangerous molds. And there are parasites, a wide-ranging group that includes single-celled protozoa and large multicellular worms. Each type of pathogen requires a different strategy from the immune system. But not all microbes are enemies. Many are part of the human microbiota, the vast community of microorganisms that inhabit our skin, mouth, gut, and other surfaces. These microbes form a protective shield, competing with pathogens for space and nutrients. Some produce vitamins. Others train our immune system, teaching it tolerance and restraint. Picture the gut lined with millions of bacteria living in harmony. The immune system watches carefully, but instead of attacking, it allows the community to flourish, recognizing that their presence strengthens rather than weakens the body. This balance between tolerance and defense is one of the immune system's most intricate yet delicate tasks. When pathogens do invade, they often exploit different routes of entry. Some will spread through the air, reaching the respiratory tract. Others enter through food and water, finding their way into the gastrointestinal system. Still, others spread through direct contact or through blood. The immune system must adapt its defenses depending on the doorway the invader chooses. As you drift further into calm, imagine the invisible world that surrounds you. Billions of microbes, most of them benign, some dangerous. The immune system is always listening, always deciding which ones to ignore and which ones to fight. It is your ultimate guardian of balance. Part 4. Barriers and first defenses. Before an invader can meet an immune cell, it must first pass through your outer body's defenses. Imagine standing at a wall of a fortress. These walls are your skin and mucosal surfaces, the body's most ancient and constant guardians. The skin is tough and layered, a physical shield that covers the body. It's dry, slightly acidic, and constantly renewing itself, shedding microbes along with dead cells. Oils and secretions add chemical defenses, making it a harsh environment for most pathogens. But the skin is not the only barrier. Inside the body, where tissues meet the outside world, lie the mucosal surfaces. In the respiratory tract, the digestive system, and the urogenital tract. These surfaces are softer, more delicate than skin, but they are coated with a powerful protector, mucus. Specialized goblet cells continuously secrete this mucus, forming a sticky layer that traps microbes before they can reach vulnerable tissues. In the airways, tiny hair-like cilia sweep the mucus upwards, carrying trapped microbes out with every breath. In the gut, thick layers of mucus prevent bacteria from reaching the epithelial lining. Even in the nose, goblet cells coat the passages, with protective layers. These barriers are more than passive walls. They actively defend. Enzymes in saliva and tears break down microbial walls. Acid in the stomach destroys ingested microbes. Antimicrobial peptides secreted onto skin and mucosa disrupt microbial membranes. Together, they form a living, layered shield. As you continue to rest, imagine these surfaces working together in harmony. The skin holding firm, the mucus flowing gently, the cilia brushing softly, all moving in harmony. Long before an invader meets a macrophage or lymphocyte, he must first try to breach these walls. And for most microbes, the journey will end here, stopped at the very edge of the body. Part 5. Innate versus adaptive immunity. Now that the barriers have been crossed, the body will now call upon deeper defenses. These come in two great forms: innate immunity and adaptive immunity. Imagine them as two wings of the same guardian army, one that's fast and older, and one that's slower but more precise. Innate immunity is the first responder. It is always present, ready to act within minutes of an infection. Innate defenses include phagocytes like neutrophils and macrophages, natural killer cells, and the complement system. Their actions are so rapid, but they are not tailored to any single invader. They respond the same way to many different threats. Think of them as a general alarm system, strong, immediate, but without much memory. Adaptive immunity, by contrast, takes some time to awaken. Days may pass before it's fully active, but once it's engaged, it provides a defense unlike any other. Specific, targeted, and able to remember. Adaptive immunity is carried out by lymphocytes, like B cells and T cells. B cells will produce antibodies that bind only to their unique antigen. T cells recognize specific peptide fragments presented on other cells. Together, they build a defense that fits the invader like a key to a lock. The hallmark of adaptive immunity is memory. Once you're exposed to a pathogen, the adaptive system remembers it. On second encounter, the response is swifter, stronger, and often stops infection before it takes hold. This is the principle behind vaccination, which teaches the adaptive system to recognize an invader without causing illness. But neither system is sufficient alone. Without an innate immunity, the body would be overwhelmed before the adaptive system could respond. Without adaptive immunity, infections would linger, never fully cleared. The two are deeply intertwined. Innate signals help activate adaptive responses, and adaptive cells in turn enhance innate functions. Now breathe slowly. Imagine these two wings working together. Innate immunity like a shield that rises instantly. Adaptive immunity like a finely trained archer who remembers every battle and target. Together, they form the complete story of defense. Part 6. Hematopoiesis and blood cells. Imagine stepping into the bone marrow, the hidden workshop inside your bones. Here, all of the blood's cellular components are created. This process is called hematopoiesis, the birth of blood cells. At the heart of this workshop are hematopoietic stem cells. These rare, remarkable cells have the power to renew themselves and to give rise to every type of blood cell. From them come the red blood cells that carry oxygen, the platelets that heal wounds, and many white blood cells that protect against infection. In medicine, we can even transplant these stem cells through a bone marrow transplant to restore immunity. As development unfolds, the site of hematopoietic shifts. In a fetus, blood is first formed in the yolk sac, then the liver, then the spleen, before settling permanently in the bone marrow after birth. From this point onwards, the marrow will become the lifelong source of blood for this child. The blood is a mixture of cell types: red blood cells, small and biconcave, carrying oxygen through hemoglobin. Then platelets, even smaller, help clot blood and close wounds. But for immunity, what we're concerned about are the key players, which are white blood cells, otherwise known as leukocytes. White blood cells come in two great lineages. The first lineage is the myeloid lineage, which produces most of the cells of the innate immune system. Those cells will include granulocytes, like neutrophils, eosinophils, and basophils, as well as monocytes and their descendants, macrophages, and dendritic cells. Then you have the lymphoid lineage, a separate lineage which on the other hand produces the cells of adaptive immunity, like B lymphocytes and T lymphocytes. It also gives rise to natural killer cells or NK cells, which belong to innate defense. Among granulocytes, the most abundant are neutrophils. They are short-lived, surviving only a few days, but they are fast, highly mobile, and lethal to microbes. They are the first responders to acute inflammation, killing bacteria by phagocytosis. Think of them as literally the paramedics for your body. When they die in large numbers, they form the creamy substance we know as pus. Eosinophils, with their bilobed nuclei, are specialized for fighting parasites. Basophils, though rare, and their tissue-resident cousins, the mast cells, release histamine and mediate allergic reactions. Together, these granulocytes form the frontline chemical and cellular responders. Now, as you breathe slowly now, imagine the bone marrow as a vast, glowing workshop, producing streams of cells, red cells flowing outward with oxygen, platelets drifting like tiny healers, and white cells preparing for their many roles in defense. The immune system's diversity begins here, in the marrow of your bones. Part 7. Monocytes, Macrophages, and Dendritic cells. From the bone marrow's steady workshop, more specialized defenders emerge. Among them are monocytes, macrophages, and dendritic cells, each with unique talents, but together forming a vital bridge in immunity. You see, monocytes, they circulate in the blood, quietly waiting. When they migrate into tissues, they transform into macrophages, large, long-lived guardians. Imagine them as sentinels embedded in every organ, patrolling for signs of damage or invasion. So if you think about it, imagine a Pokemon that's in its first evolution as the monocyte. And then over time, when they migrate into the tissues, they transform and evolve into the macrophage. Now, they are versatile. They engulf microbes, debris, clear away dead cells, and release cytokines that summon reinforcements. Some macrophages can live for months, watching over their tissues patiently. Macrophages rely on specialized receptors to sense their environment. These receptors trigger phagocytosis, the engulfing of pathogens, and the secretion of signaling molecules that alert the rest of the immune system. In this way, macrophages are not just cleaners, but also communicators, shaping the immune response. Standing nearby are the dendritic cells, with their branching, tree-like arms. They are perfectly shaped to capture antigens. Their description can be given away by their name, dendritic. When you see a neuron cell, the dendrites are like the branches that are going off of the head of the cell. Now, dendritic cells, they too can phagocytose invaders, meaning engulf them. but their most important role is presentation. After gathering antigens, dendritic cells travel to lymph nodes, where they display these fragments on their surfaces. By presenting antigens on MHC molecules, they teach T cells what to recognize and fight. This discovery was so profound that it earned recognition at the highest level. In 2011, not too long ago, the Nobel Prize in Physiology or Medicine was awarded in part to Ralph Steinman, who first described dendritic cells and revealed their role in adaptive immunity. His work showed that without dendritic cells, the innate and adaptive systems could not connect. Together, monocytes, macrophages, and dendritic cells are the great bridges of immunity. They patrol, they clean, they signal, and above all, they link the rapid innate response to the memory-rich adaptive response. As you continue to rest, imagine a macrophage quietly clearing debris from a tissue, or a dendritic cell gently carrying an antigen to a lymph node. They are not loud and aggressive warriors, but patient guides, precise and careful, ensuring that defense is specific, coordinated, and enduring. Part A. Lymphocytes and adaptive cells. Now, let us turn to the most specialized defenders of all, the lymphocytes. Unlike the rapid responders of innate immunity, these cells are patient and deliberate. They carry the gift of specificity and memory, making them the architects of adaptive defense. There are three main types of lymphocytes: B cells, T cells, and natural killer cells. Each begins its life in the bone marrow, but their paths soon diverge. B cells remain in the bone marrow to mature. Their signal is the antibody, Y-shaped proteins that recognize and bind specific antigens. When a B cell encounters its matching antigen and receives help from a T cell, it transforms into a plasma cell, producing thousands of antibodies every second. These antibodies neutralize toxins. They'll block viral entry and coat bacteria, making them easier to engulf. Some B cells become memory cells, ready to respond instantly if the same pathogen returns. T cells leave the bone marrow and travel to the thymus, where they undergo strict training. Those that survive become either helper T cells or cytotoxic T cells. Helper T cells are the conductors of the immune orchestra, releasing cytokines that activate B cells, macrophages, and other T cells. Cytotoxic T cells, on the other hand, are executioners, patrolling for infected or cancerous cells and triggering them to self-destruct. Another group, the regulatory T cells, provide restraint, preventing responses from going too far. Then there are the natural killer cells, or NK cells. These belong to the innate family, but resemble lymphocytes in form. They recognize and destroy cells that lack the normal self-markers, such as virus-infected or tumor cells. Unlike T cells, they do not require prior sensitization. They strike quickly, adding an extra layer of surveillance. Antibodies themselves carry out diverse effector functions. They can neutralize microbes directly, coat pathogens in a process called opsonization, or activate the complement system, which marks invaders for destruction. Through these mechanisms, a single plasma cell can influence an entire battle, As you breathe in and out, picture these lymphocytes as artisans of defense. Each one is unique, carrying receptors unlike any other. Most drift quietly, never encountering their match. But when the right antigen appears, they awaken. Precise, powerful, and unforgettable. Part 9. The organs of immunity. Imagine the immune system not only as cells drifting in blood, but as a network of dedicated places, organs, where immune cells are born, trained, and deployed. These are the primary and secondary lymphoid organs. The basic architecture of defense. The first is the bone marrow. Deep within bones, it is the cradle of hematopoiesis, where all blood cells arise from stem cells. B cells mature here, and many innate cells are released into circulation from this hidden workshop. The second is the thymus, a soft organ lying just above the heart. Here, immature T cells undergo strict education. They are tested to ensure that they recognize self-MHC, yet avoid reacting against the body's own tissues. Only a small fraction survive this training, emerging as functional helpers or cytotoxic T cells, the best of the best. Beyond these primary organs lie the secondary lymphoid organs, where immune responses are coordinated. The lymph nodes, small bean-shaped structures scattered along lymphatic vessels, act as checkpoints. Here, dendritic cells bring antigens from infected tissues. Naive lymphocytes continually circulate through the nodes, scanning for their matching antigen. When they meet, activation occurs, and an adaptive response begins. The spleen is another such hub. Unlike lymph nodes, it surveys the blood directly. It filters out old red blood cells, and its white pulp is rich in lymphocytes that respond to blood-borne pathogens. Scattered across mucosal surfaces are tissues collectively known as MALT, M-A-L-T, or mucosa-associated lymphoid tissue. These include the tonsils, Peyer's patches in the gut, and other clusters. They guard the entry points where most pathogens arrive, ensuring that mucosal immunity is strong. Together, all of these organs form a map of immunity. Bone marrow and thymus as the training grounds, lymph nodes and spleen as the command centers, and MALT as border outposts. As you exhale slowly, imagine walking through this network. The marrow humming with new life, the thymus testing its students, the lymph nodes bustling with meetings, the spleen filtering the blood, and the mucosal tissues watching every gateway. A hidden geography spread throughout your body, always active and always prepared. Part 10. The flow of lymph and immune surveillance. Picture a vast, gentle river system inside you. But instead of water, it carries clear lymph. Unlike blood, it has no heart to pump it. Instead, it drifts slowly, moved by your breathing, your muscle movements, and the rhythm of tiny one-way valves guiding it always forward. This river collects excess fluid from your tissues and returns it to the bloodstream, preventing swelling, keeping balance. Yet, it is more than plumbing. It's also a surveillance network, carrying antigens and cells towards the checkpoints of the immune system. Along this journey, lymph nodes sit like small harbors. Into them flow antigens from the tissues via afferent lymphatic vessels. At the same time, naive lymphocytes arrive through specialized blood vessels called high endothelial venules, or HEVs. These venules allow lymphocytes to slip from the blood into the nodes, where they mingle and search for the antigens presented by dendritic cells. If the correct match is found, activation occurs. After their stay, lymphocytes leave the node through the efferent lymphatic vessels, efferent being different from the afferent lymphatic vessels. When they enter the node through the efferent lymphatic vessels, they re-enter circulation and continue their patrol. This constant recirculation means lymphocytes are always sweeping through different nodes, ensuring that no pathogen slips past unnoticed. All lymph ultimately merges into the thoracic duct, returning to the blood at the left subclavian vein, uniting the two systems. In this way, lymph weaves through together from the tissues, organs, and the bloodstream, ensuring continuous monitoring. Now try to imagine this flow. Let your breath follow it. Slow inhales, drawing lymph forward. Slow exhales, letting it drain into stillness. Picture naive T cells drifting calmly through the current and just quietly scanning, waiting for the moment when they will be called into action. The lymphatic system is a quiet river of watchfulness, invisible, unceasing, and deeply protective. Part 11. The first line within. Imagine a quiet fortress that never sleeps. The moment a microbe attempts to enter, defenses stir. Some are simple, almost invisible, mucus flowing across the airways, carried by tiny hair-like cilia that sweep dust and microbes out like gentle brooms cleaning the corridors of the lungs. Within the mucus, secret guardians await. Defensins. Defensins are small peptides, only 30 to 40 amino acids long. They are amphipathic in nature, which means that they're part water-loving and part fat-loving. They can slide into microbial membranes, puncturing them with delicate pores. To bacteria and fungi, it is as if the wall of their shelter suddenly dissolves. The invader cannot survive. Defenses also do more. Some defensins can denature toxins, unfolding their harmful shapes so they no longer bind to our cells. Like skilled healers, they neutralize poisons before they cause harm. Flowing alongside defenses in the bloodstream is another great system, the complement cascade. Now the complement cascade, this consists of what are soluble proteins made in the liver, drifting quietly in the plasma in inactive form. Think of them like musicians holding instruments, waiting for a conductor signal. Now the moment infection arises, complement proteins activate. They jump into action, cleaving and binding in a precise order. One fragment, C3b, attaches itself to the surface of a pathogen, tagging it for destruction. Another one, C3a, floats free, calling immune cells with signals of alarm. This is opsonization and inflammation, the language of complement. As the cascade continues, late components assemble into the membrane attack complex, or the MAC. It's a circular pore that punctures the pathogen's membrane, as if a hole were drilled into its hull. Water rushes in, balance is lost, and the microbe perishes. The beauty of complement is its balance. It's always present, ready, but regulated by proteins like factor H, I, DAF, and MCP. to protect host cells from friendly fire. Without this regulation, the river of complement could turn on us. As you rest, imagine this layered protection at work. Cilia sweeping, mucus flowing, defensins piercing, complement cascading. All automatic guardians that never ask for a thank you. They are the quiet heartbeat of innate defense. Part 12. Keeping the cascade in balance. The complement system is powerful, but just like fire, it must be contained. Imagine it as a flowing river of sparks. If it's left unchecked, it could burn not only the invader, but the very forest it seeks to protect. So, let's talk about built-in safety. Proteins such as factor H and factor I act like river stones, slowing and redirecting the current of complement activation. What they do is that they reduce the amount of active C3 convertase, preventing runaway amplification. Another thing to consider is DAF, or decay accelerating factor. It sits on the surface of our own cells, pushing apart the pieces of C3 convertase, so it cannot damage the host. Alongside it, MCP, or membrane cofactor protein, will invite factor I to cut and inactivate C3b. These are molecular shields, decorating our cells with protective charms. Another protector, CD59, blocks the final assembly of the membrane attack complex, ensuring that the deadly pores do not form on self cells. But what about when regulation fails? Then what is to be done? What happens when these shields are missing? Without CD59 and DAF, the complement cascade can mistakenly target red blood cells, punching holes in their membranes. This leads to paroxysmal nocturnal hemoglobinuria, or PNH. Quite a long name. It's a rare disorder where red cells burst during the night, releasing hemoglobin into the urine. Patients end up waking up with dark urine, a reminder of the complement's unchecked power. This shows us that the immune system's strength is inseparable from its restraint. So, this leaves us with a delicate equation. Complement is not merely about attack. Fragments like C3a and C5a are what are known as anaphytoxins, summoning inflammation. They dilate vessels, recruit cells, and in high amounts can even trigger systemic reactions like anaphylaxis. Too little complement and infections will run wild. Too much, and our own tissues burn with unnecessary fire. As you drift now, think of your body as a garden. Complement is both the gardener's shears and the irrigation, cutting away what is diseased, while nourishing defenses. Always balanced, always regulated, lest the garden itself be harmed. The next 3 parts are going to be a gentle recap of everything we learned today, so we can imagine ourselves back in time, easy to visualize, and remember everything, while generating new ideas. Try your best to rest, and don't worry if you fell asleep. The rest will be here tomorrow. Part 12a. Stories of the first vaccines. Now, as you're resting here, let's drift back into time to the late 1700s in the quiet English countryside. A physician named Edward Jenner, as we mentioned earlier, had noticed something curious. Milkmaids who worked with cows and sometimes called cowpox, a mild illness that caused a few sores on the skin, seemed to never fail ill with the deadly disease of smallpox. Well, smallpox was devastating. It scarred the skin, blinded eyes, and claimed the lives of millions across the world. But Jenner wondered, could cowpox somehow protect against smallpox? Well, in 1796, he tested his idea. He gently took material from a cowpox sore and introduced it into the arm of a young boy, James Phibbs. The boy recovered from cowpox, mild and harmless, and eventually he was later exposed to smallpox. Well, when he was, it turns out he did not fall ill. This was the beginning of vaccination itself, the word drawn from the Latin word vacca, meaning cow. A simple observation that changed human history and earned Jenner the name the father of immunology. Now, let the story carry you forward to the mid-1800s, the laboratory of Louis Pasteur in France. Pasteur believed that microbes cause diseases, and that by weakening them, one might train the body to fight back. He called this process attenuation, growing a microbe under harsh, unfavorable conditions, so that it lost its power to cause harm, but still carried its identity, its signature. In 1885, Pasteur used this idea to develop a vaccine for rabies, a disease that had once seemed unstoppable and terrifying. His work gave hope, and a foundation for vaccines as we know them today. And besides Pasteur, another great mind at work, Robert Koch, a German physician, who carefully identified the microbes behind tuberculosis, anthrax, and cholera. He developed methods to grow bacteria on solid agar plates, allowing the invisible to become visible, and proving beyond doubt the link between pathogens and disease. These three figures, Jenner, Pasteur, and Koch, each carried a piece of the puzzle: a boy saved from smallpox, a weakened virus tamed into protection, and a germ revealed under the lens of a microscope. Together, they laid the foundation for vaccines and immunology, and their legacy would one day help eradicate smallpox entirely, a victory declared by the World Health Organization in 1979. As you breathe gently now, imagine the relief of generations past, the hope that spread through villages and cities as humanity slowly learned to turn nature's fiercest threats into lessons of protection. Part 12b, Everyday Immune Encounters. Tonight, let's imagine the immune system not in the pages of a textbook, but within your own body, working from morning until night. You wake up in the morning and take a breath of cool air. Invisible particles flow into your nose. Some are harmless pollen grains, unless you're allergic to pollen, and others are microbes from the outside world. Here, in the lining of your nose, tiny goblet cells release a coating of mucus. It's sticky and thick, catching dust, viruses, and bacteria. Beside them, fine cilia wave rhythmically, like oars on a gentle boat, sweeping trapped microbes outwards. Most are cleared before they ever reach deeper tissues. At breakfast, perhaps a bite of fruit carries a few unfamiliar microbes. They pass into the stomach, but here strong acid awaits, and enzymes unfold the proteins of would-be invaders. Only the hardiest can survive this path. Deeper still, in the intestine, the immune system faces a delicate balance. Billions of commensal microbes form your microbiota, living peacefully on the mucosal surface. They help break down food and train the immune system. Your body must tolerate those helpful guests, while standing guard against dangerous pathogens. To help, Peyer's patches, or clusters of lymphoid tissues in the gut wall, Survey antigens arriving with every meal. If danger is sensed, immune cells gather, ready to raise an alarm. Later in the day, perhaps you get a small paper cut. Through the broken skin, bacteria slip inside. Within moments, neutrophils that are stored in the bone marrow are released. They roll along the vessel walls, squeeze between cells, and swarm the cut. They engulf bacteria with antimicrobial granules, and their sacrifice fills the wound with the creamy material we call pus. Soon after, macrophages arrive, longer-lived, deliberate cells. They clean up the debris, secrete cytokines to call in reinforcement, and then they display fragments of bacteria like flags to signal the adaptive immune system. Elsewhere in a lymph node near the arm, a dendritic cell has carried a bacterial fragment from the cut. It presents this antigen to a naive T cell, which has entered the node through a high endothelial venule, or HEV. The T cell, if its receptor matches, awakens. Some become helper T cells, secreting signals that direct B cells to produce antibodies. Others become cytotoxic T cells, trained to kill infected cells directly. Within days, the adaptive immunity army expands. B cells differentiate into plasma cells, their inner machinery swelling as they pour out antibodies. Antibodies neutralize toxins, coat bacteria so they're more easily devoured, process once again called opsonization. And in time, the infection is cleared. Meanwhile, in the bloodstream, complement proteins are circulating silently. When triggered, they cut one another in a cascade like falling dominoes. Fragments of C3b coat bacteria marking them for destruction. C5b, C6, C7, C8, and C9. They all come together to form the membrane attack complex, or MAC, a pore that punches through a bacterial membrane, ending its life. Can you believe that all of this happens without your conscious awareness, while you sip water, answer emails, and lie down to rest? By the evening, as you drift towards sleep, the immune system remains awake. It's not done its job yet. Natural killer cells patrol the body, examining cells for signs of viral infection like security. They do not need prior memory, only the instinct to strike when something feels off. Regulatory T cells circulate too. Their role is much more subtle, to calm the immune orchestra when danger has passed, to prevent autoimmunity, to remind the system not to attack the self. And so, across the span of a single ordinary day, your immune system has tolerated friendly microbes in your gut, cleared pollen and dust from your airways, healed a cut with layers of defense, and stayed alert for infections or tumors. Most of this unfolds subtly as a tireless dance of cells and signals woven into the fabric of life. Now try to breathe slowly now, and imagine that you can imagine these guardians not as warriors in constant battle, but as caretakers, balancing vigilance with restraint, ensuring that every day, even the most quiet ones, your body remains whole. Part 12C. The Future of Immunology. As you're lying down now, imagine yourself not in the past with Jenner or Pasteur, but looking into the horizon of tomorrow. You see, the story of immunology is still being written. In the past century, vaccines erased smallpox and nearly eradicated polio. But today, scientists dream of more. New vaccines are being designed not only for viruses and bacteria, but for cancers and even chronic diseases. Some use fragments of messenger RNA, tiny instructions that teach your cells to briefly make a harmless protein so your immune system can practice without danger. Others use viral vectors or carefully engineered proteins, The goal is always the same: to prepare memory, to make the adaptive immune system ready before the real threat ever comes. In the field of cancer, the immune system is being asked to do something bold: to recognize our own cells that have gone astray. Monoclonal antibodies, once experimental, are now amongst the fastest growing classes of medicine. They can mark tumor cells for destruction, or block the signals that tumors use to hide. Other therapies release the brakes on T cells, so they attack cancers more forcefully. These are called checkpoint inhibitors. And then there are CAR T cells. Patients' own T cells removed, genetically engineered with a new receptor, and then returned to the body as a living drug. For some leukemias and lymphomas, these therapies have brought remission when nothing else could. It's as if the immune orchestra is being given new instruments, new melodies of recognition. Beyond cancer, researchers look at the microbiome, the community of microbes in our gut, on our skin, throughout the body. Where once these were invisible, now sequencing reveals them as a vast ecosystem. Some species promote tolerance and calm, others nudge inflammation. Future medicine may not just be pills or vaccines, but careful nurturing of microbial communities, restoring balance when it's lost. There is also a frontier in autoimmunity and allergy. How can we calm the immune system when it mistakenly attacks itself or overreacts to pollen or food? Biologic drugs, highly specific antibodies, already target molecules like TNF in rheumatoid arthritis, or IgE in severe asthma. In the future, therapies may retrain immune tolerance more deeply, teaching the system to recognize self as self again. And perhaps one of the greatest frontiers of all, immunology at the level of prevention and lifestyle. We are learning that sleep, diet, exercise, and stress can tilt the balance of immunity. Soluble fibers feed good microbes. Restful nights allow NK natural killer cells and T cells to function well. Even gentle movement like a walk supports lymph flow through its one-way valves. So as you close your eyes tonight, know that your immune system is not only an ancient but ever-changing. It remembers Jenner's cowpox. It adapts to each day's encounters, and is being shaped by discoveries still unfolding. Tomorrow's immunology may bring vaccines for diseases once thought impossible, immune therapies that cure cancers, and treatments that restore balance where it was broken. But tonight, all you need to do is rest, while your immune system, tireless, carries on its watch. Part 13. A gentle recap of the immune symphony. Let's go through one final recap of everything we discussed. As you settle deeper into rest, let's look back over the path we've walked. We began with the origins of immunology, Edward Jenner's cowpox experiment, Louis Pasteur's vaccines, Robert Koch's discovery of microbes. These moments were like sparks that lit the lantern of modern medicine, guiding us towards the idea that the body could be trained, its defenses sharpened, through exposure and memory. Then we explored immunology in medicine, allergies, where the immune system overreacts to harmless pollen, autoimmunity, where it mistakes self for enemy, the challenges of transplantation, and the promise of cancer immunotherapy, and the timeless struggle against infectious disease. We met the microbiota, the helpful companions that live upon and within us, as well as the diverse pathogens, bacteria, viruses, fungi, and parasites, that test our defenses. Between friend and foe lies the immune system's most delicate task, to discern wisely. From there, we saw the first lines of defense: the skin, mucosal surfaces, and secreted barriers like mucus and enzymes forming an ever-present shield. We compared innate and adaptive immunity, the innate system, fast, non-specific, and always ready. The adaptive, slower at first but precise, with the gift of memory that underlies vaccination. Together they form a duet, each strengthening each other. Earlier, we descended into the bone marrow workshop, where hematopoietic stem cells gave rise to all the cells of blood. We watched neutrophils rush to fight, eosinophils and basophils tackle parasites, mast cells release histamine, macrophages and dendritic cells present antigens, and lymphocytes, B, T, and NK cells carry out precise strategies of defense. We then walked through the immune organs, the bone marrow and thymus as primary schools of immunity, the spleen, lymph nodes, and MALT, as bustling hubs where antigens and lymphocytes meet. Then we traced the flow of lymph, carrying both fluid balance and immune surveillance, guiding lymphocytes through their constant migration. Finally, we saw the immediate innate defenses, mucus and cilia clearing airways, defensins puncturing microbial membranes, and the complement cascade, a chain of reactions of proteins tagging, inflaming, and destroying invaders. We ended by seeing how complement must be carefully regulated, with guardians like factor H, DAF, and CD59 ensuring balance, and how failure of regulation can lead to conditions like PNH. As we think about all these things, I want you to breathe in and out and be able to have a restful night as you fall asleep. Breathe in and breathe out. Let your jaw settle, let your chest settle, let your lower back settle. And imagine your feet, your body, all relaxing as the lymph spreads through your body. Thank you for listening. Good night.